Welcome to day 3 of the school. By now, we've done our warming up. We've had our little introductory things. Everyone knows how to write down a Lotka-Volterra model now. Everyone knows how to go out to the field and think about coexistence. So we're all ready to start doing all of the other things in the school. Our first lecture today will be Danny's third, where he'll be telling us stuff about interactions in co-cultures with microbial communities. Go ahead.
Good morning, everyone. Just as a quick reminder, yesterday we analyzed the two-species Lotka-Volterra model, and we learned how to use the null lines, which are the lines or the curves in other models in which one of the variables will not grow. Okay, so for example, here for this null line, the second species has 0 growth rate. And then by analyzing how these null lines cross or are displayed in the phase space, we can understand whether one species is excluding the other. So wherever you start in the phase space, you will be pushed to this stable state. We could also have coexistence, in which the stable states in which we only have one species or 0 are all unstable. And as soon as we have the two species, we will be pushed to the point where the two null lines are crossing. And also we can have bistability when there is a strong competitive interaction between the two species, and then depending on the initial conditions, either one can win. Today is going to be a mostly practical class, so there will be a couple of exercises that you will play with a little bit later. But first I want to introduce the first exercise, which is related to some work that we did recently about the multi-stability in a community of 6 species that come from nasal isolates, so from the nasal microbiota. And of course, then we will move into the exercise itself, which is to analyze the changes in competition outcomes driven by cooperative growth. So in this Lotka-Volterra model, if we have 1 or 2 species also experiencing an allele effect or cooperative growth, how these competitive outcomes will change. And another exercise that you can work on is to try to derive the equations that demonstrate that if you add a common death rate, so an added mortality to both the species, this added mortality will be pushing you in a way that that changes the competitive outcome in favor of the fast grower. Okay, which this is something that we presented yesterday, but we didn't calculate ourselves. Okay, very good. So we were interested in the bistable outcome or the multistable outcomes in part because in these systems or communities that display alternative stable states, we can have lasting changes in composition and perhaps also in the function of these communities. Here I'm presenting an example of a time series for the core groups of the gut microbiota of an individual. So scientists were monitoring the state of the gut microbiota, and these 4 groups are more or less in a stable state in terms of relative abundances over different weeks. But one day, this individual got an infection. So there is this other group that contains the pathogen, which was a Salmonella species, and it took about a couple of weeks to recover to this sickness. However, even after recovering, for many weeks, it looks like the system is very different from what we had before. So basically, what we see here is that this other group that was before at a relatively low abundance is dominating the system, and it's keeping it in that state. Okay, so it looks like the community has potentially move to an alternative stable state. But here is very difficult to understand what were exactly the mechanisms, because there are, again, many variables that can influence the health of this individual. A similar thing happened in the nasal microbiota. We have lasting changes over our life. So when we are born, the microbiota resembles the one of the vagina of the mother, the microbiota of the kid, of the baby. But then as we grow up, there are some changes in the, let's say, healthy microbiota. So in the proportion between Firmicutes, actinobacteria, and proteobacteria that we have on the nose. And in in each of these different states, we are also exposed to jumping or switching to an alternative stable state, one that is unhealthy whenever we can get some sickness, right? So there are different pathogens that we have typically, we can find typically at low abundance in the nose, but sometimes it will switch into an infection, and the abundance of these ones will go really high with the consequences for our health. So we were motivated by these switches between alternative stable states, and we will wonder whether they will happen in the lab with if we take a few species from the nasal microbiota. So this is work that I worked on with Jeff Gore at MIT and Will Lopez, a postdoc in Jeff's lab. And Will collected all these 6 different species from the microbiota of the nose. And then he did competition experiments in which he put the 6 of them together and then performed these daily dilution cycles that we were talking about yesterday to see what was the outcome. And what he saw is that starting from an equal proportion of all of the species, we will do different replicates and we will obtain very different results. In some case, one species, this blue species, has taken over the system. In other ones, we have the yellow one, or something in between that could be a different stable state or a competition that has not reached still the stable state. So to demonstrate whether we had alternative stable states here, what he did later is to start at a 95% abundance of each of these species, and the rest 5% was divided into the other 5 species, and then he will do the corresponding experiment on this. Yes?
Can you explain this first diagram again? This first, yes,
this one? Yeah. Okay, so here we put the 5 species, the 6 species together at the same proportions, okay? And then you do daily dilutions. Do you remember what the kind? Yes, okay. So they are growing in the resources, and then they are interacting in the test tube, and every day we replenish the resources with these dilutions. Okay, so after 5 cycles, this population, or this community that started here with the same abundance of the 6 species had moved into this final abundance. So basically, in 2 of the cases, we had that a large fraction of the community was dominated by these yellow bacteria. And this is another way of quantifying the same thing, actually. So this is just the relative abundance of this brown other microbe and what you find of yellow. Okay, yeah.
But how much did you dilute in each cycle?
I think 100, but I don't remember exactly then. But experiments we do are typically in between 1/30 and 1/1000. So if I'm wrong, it will be in between these numbers.
But when you start with low species abundance on the right and you dilute by a lot, you can just drive them to extinction stochastically, because...
Correct, yes, but this will hardly be the case here, because we will be transferring about 1 million individuals at least per day, and the maximum carrying capacity also at the end of the day, we will have more than billions of individuals in the test tube.
Are there replicates of this experiment?
Yeah, so each bar is a replicate. Okay, so we did here 1, 2, 3, 6 replicates, starting with this species, the Staphylococcus epidermis, at 95%. And then after 5 days, there was only Staphylococcus. And for this case, we started with 95% of SA, and we do the dilutions. And at the end, we have 100% of SA. The same here, the same here. And these two were losers, if you want. So these 4 species could dominate the community. But this one, even if it started at 95% of the community, it will lose the competition, and one of the other species that was a very low abundance will take over the system.
How do you know that? How did you conclude that even if it starts at a very low abundance, it's going to not?
This is what this graph is telling us. So here, maybe the colors are different here, and you can see it here. This CP is in this pink color. So if it will take over the system, after 5 days, we will see all pink. Same like in here, we have the blue one, and after 5 days, we have 100% of the blue one, right? But here, we started with 95% of pink, and after 5 days, we don't see pink, we see mostly this brown, and in one case, maybe the competition is still going on. Yeah? So, yes?
Replicates of this starting condition in which all of them are equally.
Yeah.
If you do replicates of that, what is the variance that you get at the end?
Here, these are 6 replicates that we started, and we ended up with 2 going here, 2 going here, and 2 going here.
So here you're starting with the same initial condition.
Correct.
You're doing the same process of dilution.
Yes.
The same environment.
Yes.
But you're ending up with the different final states.
That's right. Yeah. So this was, so of course, in the experiments, there are some stochasticity, some errors in the exact proportions that we put in, or maybe one tube is closer to the place of the incubator that is heating, like half degree more than the other one. So there are small errors in the lab and the stochastic effects, and it happened. This is an unusual result, by the way, so it will not happen for all the communities that we test, but we were surprised by this result. And because we were surprised of this, we were asking, could it be that there are alternative stable states? And by starting with this, we were close to the separatrix, where you will go one way or the other way. So to answer or to further explore this question, then we said, okay, let's now start at 95% and see if the stochasticity goes down. And that's what we can see. In most of the cases, the result now is deterministic. Yeah?
This might sound trivial, but why exactly do we dilute it like this?
Why do we? Okay, because the biological cycle of bacteria, the most usual one, will be to grow while they have resources. They might interact also during that, but then you will get into a stagnant phase. Once the resources are depleted, then you will get into a stagnant phase of the life cycle, and then there will be not so much activity, or not as much as when they are growing. Okay, so it will be a different kind of experiment. If you want to just let them there and see what happens over time, you could get into evolution as well. So it will be a different experiment. The kind that we do here is that every day they have a favorable environment, and the species are competing for a favorable environment that lets them grow. Yeah?
So if you didn't do this dilution, but just at the end of one day, you looked at the composition. You start them
right.
off all equally. And then at the end of the day, you look at the composition, no further intervention. In that case, would the final outcomes be these also?
I don't think so. we haven't done this experiment, so I cannot tell you for sure. But my understanding, based on all other experiments that I have done, is that let's say you do 30x dilution, for example, then you will grow about 5 or 6 generations in one day. So what I mean is that we start here, and let me do it just with 2 species for some, yeah, for simplicity. We start here. This is colony forming units, or the number of cells, the density of cells in the system. And for one species, we'll do something like this. And for the other species, maybe we'll do something like this. Then the population of this one is getting high, and let's say that this one is inhibiting the growth of the other one. So now it starts, as the abundance of this one is high, the effective interaction starts to be high, and maybe this one starts to be doing this, for example. But then here, they kind of reach the saturation, and in most cases, bacteria will just stop many biological functions and just hibernate. So what I will expect is that this will do this for some time. And after this, they will start dying, many of them. So many things can happen here. Maybe this one starts dying faster than the other one, or maybe they can stay for a long time like this. I really don't know. What we do in this experiment is that when we reach this point here, we dilute and we let them grow again. So I'm going to dilute by a 30x factor. And then this one is brought, let's say, here. And then this one is brought here. And you start the process again. So again, from here, this one will grow. And this one that now starts at a lower relative abundance compared to the previous day, it will grow, but very soon will start to go down. You continue this process, and after 5 days, you have that this one has gone extinct, and this is what is happening here.
But I think maybe, Danny, maybe the question was, so there is some stochasticity in which outcome you get based on which replicate you have, right? So some of that stochasticity may be coming from within cycle dynamics, and some of it might be coming from something that happens during the dilution process, right? So maybe the question was, just the first day, you prepare all the communities exactly the same way.
Okay.
So at least before you dilute, just before that, is it true that still all the 6 replicates look the same, and it's only all the stuff that happens afterwards that introduces the variability?
Okay. Sorry, I misunderstood the question. But, okay. I don't have the answer for that, because, or maybe it is, but I will need to go into either the supplementary materials or the unpublished results. I don't remember if we have the time series for everything. But if you want, I can find out. Yeah.
So, say instead of the small test tube, if you had a very big test tube, or I don't know if it can be done, but say instead of having a small one, if you had a very big test tube, and instead of replicating, if you can't allowed them for 5 cycles in the same or the time period equal to that 5 cycles, will the results be same or will it be different?
It wouldn't, because what matters is the density of cells, and not as much how many we have. So in the end, by having a larger tube, it will not change much the dynamics of the day, because during exponential growth, if they are dividing every hour, for example, even if you have a very, very large tube, you will reach there in 2 or 3 hours more, something like that. Yeah?
I was just wondering, how do you measure the abundance?
How do we measure the abundance? In this case, we take a sample and we play it on agar surfaces. I don't know, some of you might be very familiar with this, but basically what we have is a gel in a Petri dish. And then you take a sample, you dilute it so that in your sample, after dilution, you will have about 100 cells or so. And then you spread them on the agar plate, okay? So if you have two species and you have only 100 cells of those, then when you spread it, these 100 cells will be individually distributed, okay? And you will have cells from the two species. So here they will start growing, and if the colony morphologies, so the colonies that they form, are different, then you can count the relative abundance of each of the bacteria. This is hard for 6 species, but Will is a very talented microbiologist, and he looked for 6 different mediums in which only one of the species could grow. Okay, and then by that we could differentiate.
Yeah. So, so another question is, I'm assuming this is.
Maybe let's just keep all the mics on. We don't need to turn them off during the class. So.
Yeah, Danny. So I'm assuming you grew the community in some nutrient-rich media in your experiments. So I was wondering if you had a sense for what the resource or niche overlap between each of these species were, and whether they affected the competitive outcomes and these different stable states that you saw.
I don't have the answer. So if I wanted to do that, there will be 2 ways to proceed that will be complementary, I think. One is to analyze the genome and look for specific genes that you know that with this gene it should be able to feed on this carbon source, and this gene is not present in the other genome, so that will make a difference. For example, we didn't do this. We just tried a few isolates, and they were growing in this medium that people working with nasal isolates usually use. It's called brain-heart infusion medium. So it takes some animals, I think it's rabbits, but it could be whatever, and they prepare the medium from that. Okay, so one thing will be to look at the genomes, and the other thing could be let's just try a different medium compositions and try our different species and see who is able to grow in which. And usually you have to do both if you really want to understand it. Okay, yeah?
So in that example, in the first 24 hours, they will coexist like the blue and black. But after you did the dilution, so you said like, okay, black will grow, but the blue will die. So it's like in the first 24 hours, they are coexisting, but in the after dilution, is it bistability, like from coexistence? You are so I thought like blue will also grow to the same size.
But what happens is that after dilution, so here you started at the same relative abundance, and you ended up at a different relative abundance. So let's say that now this one has a 50% higher abundance than the other one. The next day, you don't start at the same initial condition. You start at one in which the black is already favored. So the black is reaching high abundance earlier than the other one, and the competition that this one feels will start earlier. And also it started growing from a lower abundance. So this is amplifying the differences in the relative abundance day after day until one goes extinct.
But yesterday, like when you saw 2 species Lotka-Volterra model, in the case of coexistence, no matter where you start,
correct,
at the end you will reach to that coexistence point.
So in that point, yes, every day you will get to the same final relative abundances. So Yeah.
Once the transient there was
Yeah, for sure, of course. Yeah, you might need 2 or 3 days, and then every day you will see that the two of them do the same thing, even if they don't end up at the very same level, each of them.
Okay, in the transient this might decrease, but at the end they will reach the same.
Yes, once, yeah. That's why we do 5 cycles, because for a competition between 2 or 3 species, usually in 5 cycles you have arrived to this stable dynamics. Yes.
How sensitive is this sort of result, for example, to the specific time scale at which you keep doing your dilutions? So, for example, if you do it every 6 hours, and that's the period when the blue species is slightly higher than the others, you could maybe have a different bistable outcome coming from the fact that you just sampled it every 6 hours instead of waiting 24 hours for the black to dominate. So do you see like qualitatively different outcomes depending on when you're doing your dilutions?
I think, yeah. So I think you can even figure out the answer from what we saw yesterday, right? So if you take the dilution as the death rate, you do a faster dilution or a strongest dilution, you are increasing the death rate. So you are going to be changing the outcome between the species. Yeah. Should I go? Yes?
In your first set of experiments, stochastic effects can take you from a stable state with multiple species to another stable state with different number of species. Whereas in this case, where you had stable states with one species, stochastic effects don't take you away from that state. Does that imply that stable states with multiple species have smaller basin of attraction compared to stable states with lesser number of species?
So is your question about whether these stable states are different than these ones?
Yeah, the basin of attraction for these stable states with multiple species, is their basin of attraction smaller compared to the basin of attraction for?
So what I would say is that the basin of attraction should be the same. We have the same community here. You start just from a different initial condition. Okay, so the stable states are what they are. You might have 4 or maybe more, but we discovered 4 alternative stable states. And then our interpretation is that here you might have started very close to an unstable fixed point or something. And by the end of 5 cycles, actually, you didn't reach one of the stable states. But I wouldn't take this one as a stable state, for example. But it's an interpretation, and of course, we needed more experiments after this. So may I continue? Yeah, good. So we wanted to know whether we could explain these 4 alternative stable states based on the pairwise competition between 2 species, right? So we took the 4 winners of the competition, the 4 that can take the whole community at the end of the experiment, and then we did competitions between them. And what we saw is bistability everywhere, which is very uncommon in our experience. So usually you will take 4 species, you will do the experiments, and in some cases you get coexistence, in some other cases you get a different thing. And here we will be competing the blue species with the yellow species, and then we will observe bistability. So depending on the initial relative abundance of that species, we will have the species winning or losing the competition. And this happened for all the possible pairs that you can do with these 4 species. Okay.
Danny, do you mind me making a comment? So this might be a bit confusing. It was certainly confusing to me the first time I encountered this, but it might seem a bit funny, this diagram that Danny has drawn on the board on the right, and all of these pictures that you're looking at from the experiments on the left, right? And I think the important point that can often get missed is that the diagram on the right on the board is kind of a zoom in of what's happening sort of all through time within each cycle. Whereas when often you're doing experiments, such as the one shown on the left, you're not tracking everything for the entire 24 hours within a cycle. Instead, what you're doing is that you're making a measurement exactly at those dotted points that Danny is sort of marking out right now. So in the data on the left, what you see is exactly just those measurements. All of the stuff that's happening within a day, that's kind of hidden, right? So when you're looking at data of this kind, and you will certainly see data of this kind, not just in this talk, but in many talks during the school, just keep that in mind. There are some internal dynamics during the cycle that are happening that you may or may not have access to. But often these kinds of experiments are easy to do exactly because you kind of leave the culture for a day or two, you come back, you make this sort of measurement, and then you just come back the next day and you keep doing this again and again. Right? So the experiment is very easy, but you have access essentially only to these dynamics at the end of the day.
Yeah.
Do you agree with that, Danny?
Of course. Yeah. Here, at the end of the day, is when we take a sample, we plate it, and then we count how many cells we have from different species. So our only data comes from this point. Thank you. Actually, that was an important fact.
So we dilute it just for adding a depth rate to maintain the.
Yes, I mean yes. So you can see like that. We add a death rate and in order to do the dilutions, but in a different way of seeing it as well is that in the Volterra model, these species, even if there is no added death rate, they will continue interacting, okay? But for bacteria, once they reach saturation, many things can happen, and maybe they just both start to hibernate and they don't continue interact in the same way, okay? So what we do is to just have these dynamics every day with a death rate every day until we reach some stable dynamics that is repeated day after day. Mm-hmm.
So in these sort of batch cultures, the environment, of course, is also changing as the food is depleted every day.
Correct. Yes.
Is it difficult to do those, continuous stirred tank, chemostat experiments with constant dilution? So if you can be sure that the environment is constant and you're looking at the attractor of the same dynamical system.
Correct. Yeah. So the question is whether we could use a chemostat where you have a constant influx of nutrients and an outflux of everything, like nutrients and bacteria, and then you see the stable state really of the system. This is not a difficult experiment, not more difficult than this one, but what is difficult is to get into high throughput, into trying many conditions. So here, what we have is that each well of a plate, a little well like this, and we have 96 wells in a singular plate, can be one of your communities, one of your test tubes, if you want, and then every day you can be working with hundreds of these bacteria, but it's hard to have hundreds of chemostats to try many replicates, many conditions, and all of that. It's just an experimental convenience.
Is it possible to translate the results of these experiments into those fixed environment experiments, or is it?
In many cases, in my experience, qualitatively, yes, it's possible. We have done the changes with a continuous death rate instead of a constant death rate, and the outcomes are the same. Of course, things change, like the exact dilution at which you obtain coexistence versus competitive exclusion, and so might change in numbers, but you can end up observing the same qualitative results. Of course, this is not going to be always true, but in many, many cases. Okay. So what we have here is a network of interactions between 4 species, and we have biostability between all the pairs, right? So between these two, if you start at a relative abundance anywhere on this line, you will fall into all CN winning or MC winning, okay? And now we have biostability between these any pair of species. And what we ask next is whether we will have competitive interactions explaining this kind of bistability. And to just further explore this, what we did is a different kind of experiment in which we grow one kind of bacteria, one species in a test tube, and we wait for 24 hours. So this species has reached the carrying capacity, has consumed the nutrients, and has secreted whatever its metabolism is secreted. Then we filter this medium and we replenish with a little bit of nutrients, but this is actually the spent media in which the other species has grown. And now we put inside the other species. So what we want to know is whether this species is going to grow worse or better in the media that the other one has used compared to when this species will grow in fresh media. Okay, so we saw a lot of bistability. What we were expecting was a lot of competition, so that many species will not grow well in the medium of other species. But this was not what we saw. Here in this matrix, we are showing the relative growth of one species when it's growing on the metabolic products of the other species. It will be red if there was a lot of inhibition, so no growth, and it will be green if actually the species was growing better than it will grow alone in this system. So one thing that shocked us is that we didn't see a lot of competition in the interspecies interactions. But another thing that we remarked is that there was a lot of self-facilitation. So this will be, if we look at the diagonal, I think that the colors are not great here, but you will see that this is a little bit red. So CN was inhibiting the growth of itself, and this is what you would expect for a logistic growth, for example. But these two are a little bit green, and this one is very green. So these species will really benefit for growing in the spent media of their own cells. And another way of seeing this is that some of the species are exhibiting an alley effect that we saw yesterday, in which the per capita growth rate initially grows with the abundance, with the density of the species, until it starts feeling the competition with peers, and of course the growth saturates. And this cooperative growth, or this alley effect, can be weak or strong, depending on whether we find an alley threshold, so a minimum density that the species need in order to start growing. Otherwise, it will go to extinction. Okay. So what we did is to now grow these species in monocultures, starting at different initial abundances, just to further prove that some of them will exhibit this kind of alley effect. And this is actually what we found. So CN was growing logistically, as we saw in the previous lectures, but then we have two species that exhibit a weak alley effect and one species that exhibit a strong alley effect. So MC will only grow and survive if it started at a high density. Okay, good. And then we started wondering what will happen if our model will incorporate this alley effect. So here we have the result for competitive exclusion based on the Lotka-Volterra model. So we have that species 1 is inhibiting the species 2 in a stronger way than species 2 is inhibiting species 1. So we have, with the Lotka-Volterra model, we will have the competitive exclusion and SE, this blue species, taking over the system. And now we wonder, okay, but what happens if this species has an alley effect? So the growth is not logistic now, but it has this quadratic term as well. How will the null class change? And one thing that can happen is that this competitive exclusion could turn into a bistability result, just because this null line now, instead of being a straight line, starts curving at low densities of the first species, and this allows a region in which the co-culture will go towards the carrying capacity of CN at the expenses of the other species. One last thing that Will did is to then try to find a way to play experimentally with this allele effect. And what he did is a bioassay in which he tried 24 different molecules that could be important for the metabolism of these bacteria, and see if some of them will inhibit the allele effect. So we'll make this allele threshold either lower or disappear. And what he found is that in this case, glutamate was a good candidate to inhibit the allele effect, because both the three species that were exhibiting the allele effect will grow better if the medium was supplemented also with glutamate. So here's what we observe. So CN, no matter if we have glutamate or not, is going to grow more or less the same. Here is how this species was growing before, and now we have glutamate in the medium, and it grows like this. So it looks also quite like this one. And here as well, the allele effect is reduced quite a lot. And for this one, it doesn't completely go away, but the allele threshold has gone to the left, so it's also reducing a little bit the allele effect. Glutamate, one single metabolite, was changing the allele effect in the three species. And the final thing that we did is to actually use glutamate in the competition experiments to have all the species there, or all the pairs of the species there, and see if now the competitive outcomes have changed. So we will go into this later, but here's where come one of the exercises that I would like you to do today. You could play to analyze different cases in which an allele effect will change the competition outcome compared to the classical Lotka-Volterra model. Okay, so just start analyzing different cases for bistability, coexistence, and all of this. When you have just the Lotka-Volterra model, or when you add that one of a species or both of the species are growing logistically. This will be one of the exercises that we are going to do. And the second exercise has to do with how an added death rate in the Lotka-Volterra model, so no cooperative growth here, no allele effect, just the Lotka-Volterra model adding a term for the death rate, and how this changes the competition outcome over this straight line of minus 45 degrees. I would like you to play with the equations to see if you can find this relationship. Just as a reminder, this is what Claire Abreu and some colleagues did a few years ago, in which they were competing two species at a different death rate. And as they increased the death rate, they were seeing how the outcome of the competition changed from one species winning, which happens to be the slow grower in here. And then here, the two species will coexist. And if we increase a lot the death rate, then the fast growing species will win the competition. Okay. And they could do this for different pairs. So instead of crossing through the coexistent phase, it will cross through the bistable phase for the species. Okay. So here are the exercises that I propose. These 45 minutes that we have now should be for you. So just choose the exercise you want, or work on the two of them if you finish, or if you prefer to work on or study anything else that we have been covering over these three lectures, just go ahead and I'm gonna be here. You can ask me questions. If somebody wants to work on exercise 1, but instead of pen and paper, you want to use your computer and draw these diagrams on the computer, the code that I used in the paper is here in the GitHub Daniel R. Amor. You will find it in the folder for the Ali effect, and then from there you can just click like run the code, and you will recover all the figures of the paper, so all of these null lines in different cases, and you can also change the parameter and see how different scenarios could unfold. So again, choose one of these 2 exercises, pen and paper or computer, as you want. If you are using your computer and you have questions, it's better maybe if you come to the first row and just start, and in a few minutes we will work on the solutions as well. Excuse me, a question about what? Sorry.
I have a question about some of the results.
Some of the results.
So you mentioned that there was a lot of bistability that you saw between pairs of when you looked at pairwise interaction.
Yes.
Now, how does that square with the initial result that you showed? That some of those very species are losers.
In the original community, we had 6 species.
Yes.
And then when we did the pairwise competition, we only got the 4 winners. So I didn't show the results for what happens in the competitions with the other 2.
So the losers were excluded.
Yes, the losers were excluded. Yeah, but also we did these competitions, and the losers were always losing, or almost always.
So there is no bistability when the pair involves the losers.
There is not. There is no bistability. Maybe with some species it might coexist, but with the majority of the winners will exclude these 2 competition, these 2 species. Not always. You will see. Maybe.
Could you go back to the picture of the losers?
Of course. Good.
CP and LP were the losers.
Correct.
LP and
CP, which will be displayed in pink color, and LP, which will be displayed in this light brown color. I think that the contrast between the colors in the projector is not great. But
is CN a loser?
No, CN is a winner. So CN is this winner, and it also won here, and it also won here. So these 2 are the losers, LP and CP.
Those are the ones that you excluded in the other.
Yes, because we were, so if we, from here, we concluded that we have these 4 alternative stable states, and we did the pairwise competition with the winners. Perhaps the data on the competitions with the other 2 are also available, but we did not present it upfront.
So in the first exercise, like we reduce the variation, so in the exercise 1, if we reduce the weekend, so it will drive the non-plan to upside. In some extent, it will stay into bistability, but I think the basin size will decrease. Again, like after a few threshold, it will reach to like only one species.
Maybe, okay. Keep it for 2 minutes. I'm just gonna write the equations that so that it's clear what kind of equations you need to use, and maybe you can come here and we can draw together what you are mentioning. Okay, just to avoid confusion, I wrote the kinds of equations that you may use. So for exercise 1, when you introduce the allele effect on one of the species, you can do it this way. So you multiply it to the logistic growth. So here I capture all the interactions within the population, within the one species. And here we also subtract the interaction with the other species. So you can play with comparing the classical Lotka-Volterra model with this model in which you have one species having the allele effect, or even if you have this form for the two species, so the two of them have the allele effect. Okay? This is one of the exercises, and the other one is to use to start from this modified Lotka-Volterra model in which we have an added mortality, and then the exercise is to try to find a change of variables that would result in recovering the classical Lotka-Volterra model. Okay, so somehow you will end up with some z, which is the new variable, and this z will grow again with a different growth rate and has this form. This will be. So the parameters will also be rescaled, but you will have incorporated the death rate into the equation. In the version that I've seen, you rescale z as a function of y, and then there are other parameters as well.
So the exercise is to find
the exercise is to
as a function of y in the parameters.
Yes.
That you end up with that.
Correct. Yeah.
So because you want your negative z1, you don't want the parameter equal to z1.
Right. Yeah. Yeah, you want it to look like it's like a Lotka-Volterra model.
But then those the other get the parameter equal to z.
True.
Right. So exactly what we want to recover. Yeah. Somehow we want to recover the normalized version of the Lotka-Volterra model so that we can plot it in the log-log space and compare it with what happens with when this is larger or smaller than 1. Yeah. Yeah. If you're very interested in this one, but you don't know how to start, you can also go into the supplementary materials of the paper there by Claire Abreu, and then you will see what is the change of variables that you have to do, and you can continue from there as well. Otherwise, I'll write it here in a moment. Yes, so I forgot to mention that you are very welcome to work together as well. So pair with your colleagues and discuss the results, discuss how to approach the problem, and also me and some other colleagues that they want, like Akshit, for example, volunteer to just hang around with you guys so you can ask us questions, we can help you in how to proceed with this, okay?
Now you want this simply easy.
So these are
Okay, for those that are working on the first exercise, here is just a little demonstration of the kinds of things that you can play with. The first thing is just to notice that when we analyze the null clines for this species, now, of course, we have the axis, y equals 0, but then the other null cline now, the other null cline now is a parabola, an upside down parabola, okay? And then in a scenario like before, in the Lotka-Volterra, we have coexistence, where we have these two straight lines that are the null clines. And then if this null cline now, which is this one, starts to transform in a parabola, we can end up in a situation like this one, in which now the two null clines don't cross, And just because of the Ali effect, we have transformed something that was a coexistence into something that will go into this stable state in which the second species win. Okay? And now, just knowing that this is a parabola, you can start playing to change the location of this parabola, the parameters of this parabola, so it could actually cross this way or the other way. It depends on whether we had here coexistence or we had bistability, and you can start analyzing the different cases and how the new stable states or the new fixed points where the null line crosses are stable or unstable. Okay. I'll show a few of them that were relevant for our work later in a few minutes. Yes.
So, once you worked out these pairwise interactions, what prevents you from using these models for a 3-species case or a 4-species case?
Nothing.
And work out what the predictions are.
So in the, I don't know if I should keep. Okay. Yes. So the predictions for communities that are relatively simple, they work pretty well. So, for example, from analyzing all the pairs of species and making trios, it works very well. You could predict 80 or 85% of the outcomes. Some of them will change because of emergent properties. And then, of course, if you move into larger communities, like let's say now you put 8 species together, maybe 60% of the times you are going to be okay with your predictions, but not always. So better than random, but it starts to be difficult. However, if you incorporate, for example, the results that you have for trios of species, and then you try to go into 8 species. This is work that Jonathan Friedman did a few years ago. I can give you the reference later. And there it could predict quite well the more complex community. But of course, then if you go into hundreds of species, then it becomes a mess. We have to use other kinds of techniques, and you are not going to be able to predict exactly the dynamics of one community, but you might be able to predict certain patterns based on the statistics of the interactions.
So, I'm concerned, let's say about this very first experiment that you talked about, you started them all out.
Yeah.
And then you went to each attractor, each replicator the attractor. 4 or 5.
Yeah.
Now,
yeah, so I think no, it's yeah, I think we have to look at dy by dt and look at the signs. Here still we are having yeah, so I think when the Ali effect is suppressed, Ali effect suppressing means reducing my one, right? I think this is reduced. But what happened when you reduce the so we had was like so basically this wait, this is weak. No, but this is this thing. This is not an alpine, no? This is the yeah. This is X-ray imperceptible. Let's take a look. I think would be changing by yeah, so basically this thing, instead of being like this, it will become like this. So it can just go up and it can come like this. Yeah. So this will be stable, this will be unstable. Yeah. But this point, I think, will depend on the yeah. Can we do it?
Yeah, so let me quickly guide you through the steps to solve the second exercise first. So the lucky idea, if you want, or after some work, the idea that they had is to write the equations in terms of this new variable that is going to be the y variable for each species, 1 minus delta over its growth rate to the power minus 1. Okay, so if you do this, I let you do the math, and you will find that you can write the new equation with this growth rate for z, which is just the original growth rate minus the added mortality. And then what you have is that the new interaction term is the previous interaction term multiplied by something. When you have this multiplication in linear scale and you take the log, you're going to have a sum of a term. Okay, so basically, if you add a mortality to one species, you will be increasing by some factor this interaction term. And this is symmetric. So somehow for the other species, you will be subtracting exactly the same term. So this means that in the log-log phase, here, whatever you add due to mortality is going to decrease for the other species. So you are going to move in a 45 angle direction. Okay. And because of this, so yesterday we already saw that for the Lotka-Volterra model, we have 4 regions in this log-log space for the interaction terms. And if we start with the species that has a lower growth rate here, then we will see that these changes will move over this line in a way that can lead us to crossing first by stability and then lead to the faster grow and winning, or alternatively to crossing coexistence and then to the fast grower winning. So making this prediction theoretically and then test it across many experiments was something that gave Claire enough work for her PhD, but also a very nice paper out of this. And then about the second exercise, we have seen that the addition of the alley effect into the Lotka-Volterra model can change the competition outcomes, and it could do it in different ways. So in order to test these predictions, what we did now is to reduce the alley effect experimentally by adding this glutamate. It's a carbon source that we'll observe that was able to reduce the alley effect or even suppress it for some species. So before we move into, okay, let me perhaps make this larger. And so when we analyze all the possible effects of changing, or many possible effects when changing, when reducing the alley effect, okay, it's going, oh, now. What we saw is that the bistability that could be driven by the alley effect, when we reduce the alley effect, could be changed to exclusion when this null line got straight. And actually is what we experimentally observed for 3 of the pairs. So when we competed CN with anybody else, as soon as we add glutamate to the system, we observed a case of exclusion where CN was excluded. Then in other cases, we had both species having a weak alley effect. Well, no, here is a strong alley effect for one of the species and a weak alley effect for the other one. This is because for these species, here is the threshold for the alley effect. So this first species is exhibiting a strong alley effect. If we shift our perspective, the crossing here should be positive if the alley effect was strong. What this species has is a negative alley threshold, and the parabola doesn't cross the axis in this way. So here we have a weak alley effect and a strong alley effect, and we observe different ways in which these change the competition after we reduce the alley effect. Reducing the alley effect didn't change much for this species, the one that has the strong alley effect, but completely reduced the alley effect for the other species. We changed this null line, and then we had a case in which bistability stayed in bistability, and another one in which we have an interesting result because we have bistability, but now this stable state is a stable state where they have the coexistence of the 2 species. So at the end of the experiment, we observed that both these species were in the system. And finally, we had a case of potential tristability. This experimentally was difficult to replicate, but we obtained it sometimes stochastically. But it looks like from the theory, when we have certain combinations of the interaction term and the alley effect, we can have tristability. And in this middle point is where the null lines will give you a stable state. Once we reduce the alley effect, what we have is a coexistence between these 2 species. And so with this, now let me come back to where I was. So somehow the network of interaction has changed. With the alley effect, we have bistability everywhere, and without the alley effect, CN was always excluded, and these were the winners. Then we had still a bistability between 2 species, and in this case we had coexistence, and in this case we had coexistence with bistability. Then we put again the 6 species together as at the beginning of our experiment, but now with glutamate to reduce the alley effect. And in this case, we when started with different initial conditions. So here we have 95% of these species at the beginning, and at the end we come here. And the same here for these other species, and at the end we come here. So in 5 of the cases, we ended up in a region for coexistence between these two species, which is arriving to this stable state. And in another case, we had a coexistence with MC, which ends up being this point here. But that was from starting with 6 species in the system. And with this, I just wanted to go a little bit through what we did today. The alley effect can drive multi-stability in microbial communities, even when this alley effect is a weak alley effect, which means that the population by itself is not bistable, but the bistability emerges once we have the competition with another species. Then we have seen as well that increased mortality favors fast-growing species under competition. And on the way during these 3 lectures, we have seen how we can use microbes that grow very rapidly and we can control in the lab and even exposed to different conditions are a good platform to match the ecological theory with experimental tests. And with this, thank you very much. It's been a wonderful 3 days spending with you.